Hello everyone, continuing with the previous discussion on pancreas from physiology point of view, and we have discussed about insulin, the synthesis of insulin. Today we are going to discuss the secretion of insulin. How is it secreted? For the secretion of insulin, it depends upon the glucose metabolism, which creates ATP. This is a cyclic AMP dependent process. So it depends upon glucose metabolism, it depends upon cyclic AMP. So let us imagine if this is a cell and these are the GLUT2 receptors, glucose will enter through them and now it will undergo glycolysis, the enzyme like glucokinase, and it is going to form ATP. By forming ATP, now there are already two channels which are present here. One is a potassium channel. This potassium channel and there is a calcium channel. Okay, and this is the storage granules which contains the insulin. Okay, this is the secretory granule. Now normally what is happening, this is going to cause the potassium efflux. Okay, it is going to cause potassium efflux. Now but with the glycolysis there is increased ATP formation, and this increased ATP formation is going to inhibit this channel, therefore no more potassium efflux, so increased potassium level inside the cell, so it causes a depolarization of the membrane and hence it will open up the calcium channels. So now the calcium will enter inside the cell. So calcium will enter inside the cell and calcium will help in the microtubule contraction and the release of the insulin outside the cell. Okay, so let us see once again. So what is happening, the glucose is entering inside, then it is going to form ATP via glycolysis, then this is going to inhibit the potassium channels, so there will be increased intracellular potassium level which will depolarize the membrane and it will stimulate, it will open up the calcium channel, voltage sensitive calcium channel. The calcium channel will cause the influx of calcium, so there will be inside there will be more calcium, and this will facilitate the contraction of the microtubule and the exocytosis of the insulin. So this is how insulin is synthesized. Okay, if we see the normal basal rate of insulin synthesis, of insulin release, actually normal basal rate of insulin release from beta cell is around 1 to 2 unit per hour. Okay, and if you take your food, it 5 to 10 fold increase happens. Okay, the normal release of insulin is 1 to 2 unit per hour, and with the ingestion of food it increases by 5 to 10 folds. Okay, now let us see the transport, how insulin is transported. Okay, so insulin will bind with the plasma proteins. Insulin binds with the plasma protein like synalbumin. Half life of the insulin is around 5 to 10 minutes. Okay, so there are insulin receptors on the cells. These are glycoprotein receptors. So insulin exert its effect by interacting with these receptors. So insulin doesn't need to enter inside the cell. It doesn't need to enter inside the cell to have its effect. It will have its effect via interacting with the receptors present on the cell membrane. Okay, now if we see the metabolism of insulin, how insulin is metabolized, 80% of the insulin is metabolized in liver and kidney. 80% of the insulin is metabolized in liver and kidney with the help of the enzyme HGIT, hepatic glutathione insulin transhydrogenase. 80% is metabolized in liver and kidney using the help of the enzyme, hepatic glutathione insulin transhydrogenase, and the rest 20% is metabolized in the rest of the body tissues, and here the insulin is degraded using the enzyme insulin proteases. So this is about the secretion, the transportation and the metabolism of the insulin. Now let us see how it is regulated. the regulation of insulin secretion. Regulation of insulin secretion. Now, in regulation, the most important control is via the substrate. So we can say the substrate control. Okay, this is the most important control. It's the most important factor in the control of insulin secretion. So herein we will see the control of control by the carbohydrates. Okay, control by carbohydrate, there is a primary response and there is a secondary response. Primary response is the immediate release of insulin whenever you take food. Immediate release of insulin, not take food, which occurs within 0.5 to 1 minute of glucose infusion. So if we infuse the glucose within 0.5 to 1 minute of infusion, there is a release of insulin. That is an immediate release of insulin. That is the primary response. Okay, and it reaches its peak in 3 to 5 minutes only, and then it declines and declines, but it remains above the basal level. It declines but remains above the basal level. So let us see once again. The primary response is the immediate release of insulin, which occurs between 0.5 to 1 minute of glucose infusion, and it peaks at 3 to 5 minutes and then declines but remains above the basal level. Okay, and how does that release? This is due to the release of insulin from a labile pool of insulin. Labile pool of insulin within the beta cells. So this is due to the release of insulin from a labile pool of insulin within the beta cells. Okay. Secondary response is a slow, maintained and a prolonged release of insulin. Slow, maintained and a prolonged release of insulin, which occurs on 2 hours of continuous, around 2 to 3 hours of continuous glucose infusion. Continuous glucose infusion. Okay, it is a slow, it is a maintained and it's a prolonged release of insulin, which happens after 2 to 3 hours of continued glucose infusion. And so we can say this is a delayed response or secondary response or the second response. This delayed response is due to the release of insulin from a stable pool. And this comprises 98% of pancreatic store of insulin. The 98% of pancreatic store of insulin is the stable pool. Okay, so we can see that if this is the insulin level, this is the basal insulin level, and now we have given the IV glucose here, there will be an immediate release of insulin. Immediate, so this is your first response. But after a few times there will be a secondary release of insulin, which will be a slow, a maintained and a prolonged release, okay, of insulin. So this will be the secondary response. Okay, so in substrate control, the first control we have seen the control by carbohydrate. The second control is by the control by proteins and fat metabolites, protein and fat derivatives. So we have a mixture of essential amino acids or basic amino acids. They are going to stimulate the beta cells and increase the insulin secretion, while fat derivatives or beta keto acids, they all are also going to increase the insulin synthesis, secretion. So they are also going to stimulate the beta cells. So both the protein metabolites, that is essential amino acids or basic amino acids, as well as fat metabolites like beta keto acids, etc., some fat derivatives, all of them are going to stimulate the beta cells and going to increase the insulin synthesis. So this is the first control we have seen, the substrate level of control, the substrate control of insulin secretion. Now we will see the second level of control is bicyclic AMP and substances which produce cyclic AMP and cyclic AMP generating substances. cyclic AMP generating substances. So here we can see that beta adrenergic receptor, not beta cell, I'm talking about beta adrenergic receptor stimulation. If we stimulate the beta adrenergic receptor, this will cause increased intracellular cyclic AMP, and hence this is going to increase the intracellular calcium level. How? Because it will activate the phosphorylate, it will the enzyme, phosphorylate the enzymes, and it will activate the glucokinase, so there will be more ATP generation, and it's going to inhibit the potassium, and ultimately it is also going to increase the calcium level intracellularly. So increased intracellular cyclic AMP is going to increase the intracellular calcium level also, and this is going to increase the insulin secretion. Okay, so directly we can say that beta adrenergic receptor stimulation is going to increase the insulin secretion because of cyclic AMP. And if we stimulate the alpha adrenergic receptor stimulation, this decreases cyclic AMP, and if it decreases cyclic AMP, this is going to decrease the insulin secretion. Okay, so under cyclic AMP we have to remember beta adrenergic receptor stimulation is going to increase insulin secretion, alpha adrenergic receptor stimulation decreases cyclic AMP, decreases insulin secretion. Now we should remember that cyclic AMP is destroyed by an enzyme known as phosphodiesterase. And there are some agents like theophylline, oral hypoglycemic agents like biguanines, that is metformin, or sulfonylureas. They all inhibits the phosphodiesterase enzyme. So if they inhibit the phosphodiesterase enzyme, they are going to increase the cyclic AMP level, and therefore they are going to cause increased in insulin level. So ultimately, if they are going to increase insulin level, they are going to cause hypoglycemia. Okay, so we need to remember that theophylline or biguanines, oral hypoglycemic agents like biguanines or sulfonylurea, is going to inhibit the phosphodiesterase, which will increase the cyclic AMP. Therefore, it is going to increase the insulin secretion. Therefore, will cause hypoglycemia. So herein we have seen two levels of control. One is the substrate control, and then we have seen cyclic AMP. Now the third one is the neural control. Neural control, we need to know about autonomic nervous system first. Autonomic nervous system we know is sympathetic and parasympathetic system. Sympathetic system decreases the insulin, and parasympathetic system increases the insulin. Okay, so you can remember this like this: the sympathetic system is a state of emergency and you need glucose for it, so we need decreased insulin level, and the vice versa for parasympathetic. Okay, in CNS, if we see, during feeding, if you are feeding, so there is an increased vagal activity. If there is going to be an increased vagal activity, we have seen parasympathetic insulin, there will be increased insulin secretion. If you are starving, there is increased sympathetic activity. If increased sympathetic activity, this is going to cause decreased insulin. So this is the neural control. Now we'll see the hormonal controls. In hormonal control, there are some GIT, gastrointestinal hormones, like we have glucagon, secretin, gastrin, GIP. All of them increases insulin. Glucagon, secretin, gastrin, GIP, all of them increases insulin. Also, exogenously, if we give insulin, exogenous insulin administration is going to decrease the endogenous insulin. Okay, growth hormone, this stimulates the beta cells, so increased insulin. Then we have somatostatin. We know it is opposite, so it is going to inhibit the beta cells, therefore decreased insulin. Other hormone like thyroid hormone, glucocorticoids, and oral contraceptives, estrogens. So all of them are going to cause beta cell exhaustion. So therefore decreased insulin secretion. We remember only growth hormone and GIT hormones are going to increase the insulin. GIT hormone like gastrin, secretin, glucagon, GIP. Then growth hormone is also going to stimulate the beta cells. Then exogenous insulin. Then we have somatostatin, then thyroid hormone, then corticosteroids, glucocorticoids, or oral contraceptives. They all cause beta cell exhaustion, therefore decreases insulin level. So these are the four control: substance, substrate level, cyclic AMP, neural control, hormonal control, and now we have one more level of control, that is drugs also interact. Drugs also control. There are some drugs like diazoxide, thiazide, phenytoin. Diazoxide, thiazide, phenytoin, all of them inhibits beta cells. They inhibit beta cell, therefore decreases insulin level. So therefore hyperglycemia is associated with thiazide, phenytoin, or diazoxide. There is another one, alloxan. Alloxan actually destroys beta cells, and therefore it is going to decrease the insulin secretion. So we have talked about dioxide, thiazide, and phenytoin. It is going to inhibit the beta cell and thus cause decreased insulin, but alloxan is going to destroy the beta cells and then decrease the insulin. So this is all about the regulation of insulin. Okay, we can revise it once more. Regulation of insulin. In regulation, I have talked about substrate level control, in which I've talked about glucose, the carbohydrate. I've talked about primary response and secondary response, then protein, and then fat derivatives, which are going to increase insulin secretion. Cyclic AMP, if increase cyclic AMP, it is going to increase the insulin. If decrease cyclic AMP, it is going to decrease the insulin secretion, in which we have heard about beta receptor stimulation and alpha receptor stimulation. Beta is going to increase insulin, alpha stimulation decrease. Inhibition of phosphodiesterase will increase cyclic AMP, and therefore it is also going to increase the insulin secretion. Then we have talked about the neural control, in which we have seen sympathetic system है अगर है autonomic में, तो it is going to decrease the insulin level, and parasympathetic है तो it is going to increase the insulin level. CNS में we have seen in case of feeding or starvation. Feeding तो increased vagal activity होगा, increased insulin, and then starving में increased sympathetic activities there. Then I've talked about hormonal control, in which GIT hormones and growth hormone are going to stimulate, rest somatostatin or thyroid corticoid or oral contraceptive are going to inhibit. Then drugs we have talked about: dioxide, thiazide, phenytoin, and alloxan. Okay, so these are the control or regulation of the insulin secretion. Okay, now we can talk about the actions of insulin. First, we can talk about on carbohydrate metabolism. On carbohydrate metabolism, insulin is going to promote the glucose uptake. Insulin is going to promote the glucose uptake into the tissues. In adipose tissue and in muscle, it increases the number of blood transporters. In case of liver, it increases the glucokinase activity. Okay, and this is how it stimulates or increase the glucose uptake by the tissues. It will increase the glycolysis. Actually ग्लूकोज कम करना है ना, तो ग्लूकोज यूटिलाइज करा दो बाई टिश्यूज और ग्लाइकोलिसिस करके उसे मेटाबोलाइज कर दो, है ना. ग्लाइकोलिसिस इन ऑल द 3 tissues, adipose, muscle and liver, 3 of the tissues. Okay, and then promote the synthesis of glycogen in liver and muscle. तो it will stimulate the glycogen synthesis, तो उससे भी ग्लूकोज कम हो जाएगा, and will decrease the gluconeogenesis. तो न्यू ग्लूकोज फॉर्मेशन नहीं होना चाहिए, एंड जो ऑलरेडी अवेलेबल है उसको या तो अपटेक करा दो, या फिर ग्लाइकोलिसिस करा दो, या फिर ग्लाइकोजन सिंथेसिस करा दो. But uptake के अंदर we need to remember the difference that in case of muscle and adipose tissue it increases the number of blood transporter, while in case of liver how it increases the uptake by increasing the activity of glucokinase. Okay. Now we can see on fat metabolism how it is affecting. So insulin will promote the synthesis of free fatty acids and triglycerides, in all muscle, adipose tissues or liver. Okay, so it is going to increase the lipid synthesis. I've talked about it is an anabolic hormone. It actually stimulates the lipoprotein lipase and inhibits the hormone sensitive lipase. Also, it is anti-ketotic, therefore it increases the uptake of ketone bodies in the muscles. It increases the uptake of ketone bodies in the muscles. In case of protein metabolism, it is going to promote the uptake of amino acids. It will decrease the protein breakdown and will increase the protein or stimulate the protein synthesis. As I told you, it is an anabolic hormone. It is going to stimulate protein synthesis as well as lipid synthesis. Okay, lipid synthesis by stimulating the lipoprotein lipase and inhibiting the hormone sensitive lipase. Okay, and it is also, so if we can see that it is glycogenic, है ना, glycogen form करता है. It is anti-gluconeogenic, है ना, ग्लूकोज नहीं फॉर्म करना है, ग्लूकोनियोजेनेसिस नहीं होना है. इट इज़ एंटी लाइपोलिटिक, है ना, हार्मोन सेंसिटिव लाइपेज को इनहिबिट कर देता है तो लाइपोलिसिस नहीं होने देगा. इट इज़ एंटी कीटोटिक, है ना, कीटोन बॉडीज का अपटेक बढ़ा देता है एंटी, और इट इज़ एन एनाबॉलिक हार्मोन. ओके, so these are the main actions of insulin, but there is some other miscellaneous actions also, that insulin will increase the potassium entry into the cells, and therefore it will decrease the potassium level in the extracellular fluid. Okay, we have seen this, that because of insulin there is increased glucose uptake, and then we can see that it increase the ATP level, which will inhibit the potassium channels. So there will be decreased efflux of potassium, तो potassium extracellular fluid में कम हो जाएगा, cells के अंदर ज़्यादा हो जाएगा. तो अगर हम डायबिटिक कीटोसिस में देखते हैं, डायबिटिक कीटोएसिडोसिस में अगर हम देखते हैं, तो देयर इज़ ऑफन हाइपोकैलेमिया. ठीक है, तो जब भी हम पेशेंट को ट्रीट कर रहे हैं डायबिटिक कीटोएसिडोसिस के अंदर, वी आर गिविंग देम इंसुलिन, है ना. तो इंसुलिन अगर हम देंगे तो इट विल कॉज मोर एंट्री ऑफ पोटेशियम इनसाइड द सेल, तो इट विल कॉज मोर हाइपोकैलेमिया, है ना. तो हाइपोकैलेमिया विल इंक्रीज, तो इसलिए हमारे को पोटेशियम शुड ऑल्सो बी गिवन अलॉन्ग विद इंसुलिन. तो दिस इज व्हाट वी हैव टू रिमेंबर, that it increases the insulin increases the uptake of potassium into the cell, therefore it decreases the extracellular fluid potassium level. So whenever in ketoacidosis there is often hypokalemia, and if we give insulin as a treatment alone, then it is going to aggravate the hypokalemia. Okay, so these are about the actions of insulin. Now let us see what is the mechanism of action of insulin. As I told you that insulin does not enter inside the cell and act. There are some receptors on the cells. Okay, so there are some insulin receptors through which it acts. Insulin receptors. The gene for insulin receptor is located on chromosome number 19. Okay, the insulin receptor are tetramers. The tetramers means they are made up of 2 alpha and 2 beta subunits. These are glycoprotein subunits. Okay, so it is a tetramer. It is made up of 2 alpha and 2 beta subunit. Alpha subunit is the extracellular subunit, and it is the alpha subunit, if it is extracellular, this is the one which is going to bind to insulin, while beta subunit is the one which is transmembranous, which will extend through the membrane, through the cell membrane. Okay, and it is the beta subunit which has tyrosine kinase activity. So what will happen? Insulin will bind here alpha subunit, so it will stimulate the tyrosine kinase activity into the beta subunit, and therefore it will lead to its autophosphorylation. So it will lead to its autophosphorylation. So it will cause the phosphorylation of intracellular proteins. It will cause phosphorylation of the intracellular protein, and through which insulin exerts it. This is how insulin exerts its effect. Okay, directly नहीं करता है, कुछ proteins का phosphorylation हो जाएगा, then they will become activated, and then this is how the insulin acts. Okay, The insulin binds onto the alpha receptors, and the beta receptors have tyrosine kinase activity, which undergoes autophosphorylation, and whatever intracellular proteins are present, they phosphorylate them. Okay, this is how insulin act. So this is what we have discussed in the secretion, the transportation, the metabolism, the regulation of insulin secretion, and the actions of insulin, and the mechanism of action of insulin. This is all. Thank you.